Hello everyone. In this video, we will discuss a few important Viva questions related to immunology. So let's start the video.
All right, your first question. What is antigen-antibody reaction?
Antigen-antibody reaction is a specific immune response where an antibody binds to its corresponding antigen. And this reaction helps in neutralization, agglutination, or destruction of the antigen.
Well, your next question. What are the types of antigen-antibody reactions used in the lab?
There are several types of antigen-antibody reaction used in the laboratory. The main one include precipitation reaction, agglutination, complement fixation, neutralization, ELISA, immunochromatography, and immunofluorescence.
Well done, girl. Well, could you differentiate between precipitation and agglutination reactions?
Yay, I know this answer. In precipitation, a soluble antigen reacts with a soluble antibody, while in agglutination, an insoluble antigen like RBCs or latex particle react with the antibody. And as a result of this reaction, precipitation leads to the formation of insoluble precipitate and agglutination result in the visible clump.
Well done. That was a clear explanation. You may go now. Hey, can you tell me what are epitopes?
Epitopes are also called antigenic determinants, and these are small specific regions present on an antigen. These regions bind to the immune components such as antibodies and T cell receptors and trigger an immune response.
Proud of you.
Tell me, which of the following is not a cell of innate immunity? Natural killer cell, mast cell, macrophage, or B cell?
First, innate immunity is the body's first line of defense, and it includes cells that act quickly and non-specifically against pathogens like natural killer cells, mast cells, and macrophages. But B cells are different. They produce antibodies, so they belong to the adaptive immunity, which is more specific and takes time to develop. So the correct answer here is B cells.
All right, next question. Can you name the primary lymphoid organs and explain why they are called primary?
Yes, I know the answer. Bone marrow and thymus are the primary lymphoid organs, and they are called primary because they are the sites where lymphocytes develop and mature. B cell mature in the bone marrow and T cell mature in the thymus.
Good. Next question. Define haptens and give one example.
These haptens are small molecules or incomplete antigens, and they are called incomplete because they cannot trigger an immune response on their own. However, when they attach to a large carrier molecule, they form a complete antigen, and this combination becomes immunogenic, meaning it can stimulate an immune response in the body. Penicillin drug is a classical example of hapten. It can bind to proteins in the body and trigger allergic reactions.
Perfect. You may go now. Hello, your first question is, which type of immunity is provided by vaccination?
Vaccination provides artificial active immunity
Good. But why it's called active?
because our own immune system is actively involved and produces antibodies and memory cells in response to the vaccine. That is why it is called as active immunity.
Very good. But why artificial?
It is artificial because the antigen is introduced from outside through a vaccine, not by natural infection.
Excellent. That means the body is prepared in advance to fight the actual pathogen. Good. Your next question. Which T cells are mainly responsible for killing virus-infected and tumor cells in the human immune system?
The correct answer is cytotoxic T cells, also called CD8 T cells. They directly kill virus infected and tumor cells by releasing perforins and granzymes.
Next question. Which cells are primarily targeted by the human immunodeficiency virus, HIV?
The answer is CD4 T helper cells. HIV bind to the CD4 receptors present on CD4 helper T cells, and these T cells are very important for coordinating immune responses. And when HIV destroys these T cells, the immune system becomes severely weakened, leading to AIDS.
Next question. Which type of immunity is transferred from mother to fetus through the placenta or breast milk in the human immune system?
This type of immunity is called passive immunity because preformed antibodies are introduced into the body. And in this case, it is natural passive immunity, where the mother naturally transfers her antibodies to the baby. Mother passes IgG through the placenta during pregnancy and IgA through breast milk after birth. And this provides immediate protection to the baby until the baby's own immune system becomes active.
Good. Next question. Which type of hypersensitivity reaction involves the formation of immune complexes that deposit in tissues and trigger inflammation in the human immune system?
It is type 3 hypersensitivity. In type 3 hypersensitivity, antigen-antibody complexes form in the blood and get deposited in tissues like kidneys, joints, or blood vessels. And these deposits trigger inflammation by activating complement system and causing tissue damage.
My last question. How do you all know these answers so perfectly?
Because we follow Laugh and Learn Microbiology with Dr. Saher.
Impressive. Very impressive.
That's all for this video. We will be back soon with part 2 featuring more Viva questions. Until then, subscribe to Laugh and Learn Microbiology, like, comment and share this video with your friends. Thanks for watching. See you in the next video.